This is part 2 of 7.1. So we're going to have a quick review again of the work that led to the discovery of DNA structure. So what was the work? Well, there were two major things. One was the work by Chargaff, and this suggested a particular ratio. There was a particular ratio found within DNA where A was always at the same ratio or the same percentage as T, and G was at the same percentage as C. And so this was very strong evidence that these paired together. So adenine and thymine, guanine and cytosine. And the other thing that they had was the X-ray crystallography. They had this picture that suggested this helical shape, and it also, from working out the sizes, they knew how far apart the rungs of the ladder were. And then they also had the composition of the bases, and they knew the structure well enough to think about where bonding might happen. So, yeah, that's just a quick summary then. Chargaff's ratio, C and G having the same, A and T having the same. X-ray crystallography, by sending X-rays through. DNA doesn't actually crystallize very well. It doesn't really form crystals, but it was good enough that they could get the shape of it, because it doesn't form regular repeating crystals, but it was using the same technique. And so this was also diffraction, because as they go through, they kind of spread out. So the diffraction caused the X-ray film to develop. And here was that picture which was produced from Rosalind Franklin's work, where you can see these kind of repeating units. You see the helical shape, and you can also work out how many nanometers apart the rungs of these ladders were. And so that's what Watson and Crick then used. So who were Watson and Crick? What did they discover? And what did their work suggest in terms of DNA replication? Well, here they are. They were two scientists who were working, hoping to discover the structure of DNA. And they were working with cardboard cutouts, using modelling in order to try and develop their structure. And they used the X-ray crystallography picture, and they used Chargaff's ratio. So in their model, they had sugar and phosphate as the backbone, with nitrogenous bases pointing inwards, and this matched their evidence. And they could also see how hydrogen bonds would link the nitrogenous bases from the two strands. Now this suggested, because it was hydrogen bonds between the two strands, it suggested semi-conservative replication, and we'll come to that. It's where if we have two parent strands, when we copy DNA, each new strand, because this is double-stranded, we need to have two double-stranded. Well, each new will have one which was from the parent and one which is newly synthesized. And that means it's semi, half conservative. Conserve means to keep, so it's half keeping. It's keeping half the original, and it's adding one new strand in. And so their model, because they could see that it was down the middle, it was hydrogen bonds holding the two strands together, which are quite weak, but it was covalent bonds along the strands. That means that then it would be easy to pull these two strands apart and to use one as a template. So our next question is about the structure of DNA, and how is DNA structured? Well, it's structured around nucleosomes. So in eukaryotes, in species that have a nucleus, we have packaging of DNA. And it's packaged up we have DNA associated with protein. So we're using protein to help package. And these proteins are histone proteins. So if we think about the fact that it's not just DNA, it's DNA and protein, we can call DNA and protein chromatin. It's DNA wound around proteins. So if you think about if you had a very long piece of string, the way that we keep string packaged is that we wind it around a reel. So we can get lots and lots of string around a reel in a very condensed way rather than just having all of the string by itself. And so a nucleosome is the version of the reel. It's a protein reel around which we're going to wrap the DNA. So here's our nucleosome structure, and you can see that we've got 1, 2, 3, 4, 5, 6, and behind it's the same thing. It's like this. We've got basically 8 balls. And so these are 8 histone proteins. So there are 8 histone proteins which come together to act like the reel to wrap around the DNA. And we've got this other protein here, this linker, which is holding the DNA on. And so a nucleosome is made out of these 2 different types of histone, one which makes up the 8 balls, and because it's 8 we can say it's an octamer. So an octamer of 8 histone proteins and 1 linker protein. And then you can see that the DNA is wrapped around, and it's almost 2 turns. It's going around almost 2 turns. It's actually exactly 1.7 turns, which is 146 bases of DNA. Wraps around, and then it goes off, and then there'll be another nucleosome. So let's just have a look at all that. So a whole nucleosome is made out of DNA. It's made out of histone proteins, and there's an octamer. So the whole thing, the nucleosome structure, is core DNA. So we have DNA wrapped around an octamer of 8 histone proteins with a histone H1, which is a linker and holds the DNA onto those other histone proteins. And so what this creates is it creates a fiber that is, so if we zoom in here we see it, we get DNA wrapped around those 8 histone proteins with a linker, and then we get them wrapping up, and we get overall, you can see it's kind of like a layer of 2. We get overall a beads on a string, and those beads on a string, what's called the beads on a string of the nucleosomes, then come together to make what's called the 30 nanometer fiber, or the 30 nanometer solenoid, you will see. So this whole thing here, that is 30 nanometers. And then what that does is that then goes backwards and forwards and backwards and forwards and backwards and forwards, and it creates an extended form of the chromosome. And so we see that this kind of goes backwards and forwards and then is arranged and arranged and kind of wrapped around until eventually we get to the X-shaped structure. Now it's important to say that these structures are only ever formed when we are copying DNA. Normally, when we're not copying DNA, we would have the kind of beads on a string structure of DNA. It wouldn't be wrapped up into these tight structures, these X shape. So only during DNA replication do we get this structure being shown, and we're packaging it up. So that's one of the functions of nucleosomes. It's to help package for copying DNA. Okay, see if you can stop the video and think about what were all of these different structures made from. So we had, as a quick review, we had DNA, then wrapping around our 8 histone proteins to make the beads on a string. Those coming together to form this 30 nanometer fiber, which then go backwards and forwards and backwards and forwards, and then we get this section of what we call chromatin, which is the DNA and protein, until eventually we get the whole chromosome form. So what's the function of nucleosomes? Well, we talked about the major one, which is that it's how we package. It's the packaging of DNA. And that packaging is going to allow for cell division. It's our way of organizing DNA. If we're going to divide the DNA, then we need to organize it so that we can get the right DNA into the 2 daughter cells. So they help to package it. The other thing they're going to do is, through this packaging, they're going to regulate its use. So what do we mean by use? Well, use of DNA is about reading the code. So we're reading the code and we make a copy. So we're making a copy, and we go from DNA, which is double-stranded, we make a single-stranded copy in mRNA that we'll have a look at when we look at transcription. So if it's very tightly wound around the nucleosomes, wound up in the nucleosome structure around the histone proteins, if it is tightly wound up, then we won't be able to use it. So the use of DNA is dependent on the packaging. And so we can't use DNA when we have packaged it up. If it's packaged into a chromosome, the only thing we can do is divide the DNA. We can't use it to read the code, and we can't use it to make a copy or make protein. And so all of these things, reading the code, making the mRNA, and making the protein, these are what's called expression of genes. So reading the code to make a protein, that's gene expression. And so nucleosomes can regulate gene expression. So here's our quick recap, and you notice that we're starting to use this word supercoiling and condensing. So to supercoil means when we have wrapped it up, and it's going to be during cell division that we get supercoiling of all the DNA. And so wrapping it up very, very tightly, supercoiling it, means that we reduce the size of the DNA. So this is condensing. And so it's about 15,000 times smaller than it would be if it was not packaged into nucleosomes. So we said the histone proteins are responsible for packaging, and the fiber, the DNA fiber, what we said was, so when we package it in, it's called chromatin, the histone proteins and the DNA. And that chromatin fiber must be less condensed for the use of DNA, for transcription to occur. And so how condensed it is controls if the genes are expressed or if they're transcribed. So the process of, so this is expression. Expression of genes is transcription. And if it's supercoiled, then we can't use it. We can't cannot be transcribed, it cannot be expressed. There's no expression of genes. What is the difference between coding regions for genes and repetitive sequences of DNA? Within the DNA, there are regions which we call coding regions, and this means they have information for proteins. So there's some different definitions of coding, but let's think that we're thinking about regions that make protein. So there are regions in the DNA which have the code to make a protein. If we looked along the genetic code, whatever it is, then if we took a section of it, then particular sections would have information to make a protein. And what that means is that other sections, this section over here, does not have. This is not the information to make a protein. And so this region here would be a coding region, because we can make a protein, and this would be a non-coding region. And so when scientists have analysed DNA in humans, we found that actually there's a lot of our DNA which does not have the information to make protein. And it is non-coding, and sometimes, well, quite a lot of it is repetitive sequences in the DNA. And we're going to have a look at what those repetitive sequences are like. So if we think about chromosomes as being one long, they're a linear strand. Now, actually, this is double-stranded along the whole way, but we're just going to represent it as a single strand. That means that we could kind of draw regions where we have genes, and then we would have regions where we didn't have genes. So I'm just drawing little boxes. There aren't. It's not actually a structure, it's just a code. And so there are a different number of genes on each chromosome. The longer the chromosome is, the more genes it has, but we also have all these regions in between, and these are non-coding. And these can have repetitive sequences in them. Sequences that just repeat. And this is what we can use for DNA fingerprinting, because they're unique to each individual. So here's the first example of some of these repetitive sequences. So these are called STRs. They are short tandem repeats. Tandem, like a tandem bike, a tandem bicycle is where you go together, you ride together. And so these sequences are together. So let's have a look what's happening here. We've got this sequence which repeats. Gatter, 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 gatter. And for the first person, when we sample them, sample 1 DNA, there are 8 repeats. And then for person number 2, there are 9 repeats, and person number 3, there are 10 repeats. So in different people, in between the genes would be either side. In between the genes, we get this non-coding. It doesn't make protein. And it's these repetitive sequences. So the first one, STRs, you can see they're very short. Here we've only got 4 bases repeated. 4 nucleotides repeated. Gatter, gatter, gatter. And so these repetitive sequences in the DNA can also be called satellite, because they're kind of outside of the coding region. And the second type, or the second name, is variable number tandem repeats, because the number in these varies, and these are larger. So these are larger repeated. And you can see here it's saying it's hundreds of base pairs repeated 4 times. The definitions vary, but in many it's kind of beyond 10 bases is called a VNTR. And if we're thinking about short tandem repeats, then 2 to 6 base pairs. So that was the previous one we looked at with a repeated sequence like GATA. And so that's an STR, GATA, could be repeated lots of times. And our VNTR is usually defined as beyond 10 bases up to hundreds of bases. So what do these sequences do? Well, scientists have tried to look at that, and we found that if we cut out these sequences, then our DNA doesn't work. So it has a structural role, certainly a structural role, and some of the non-coding DNA is also acting like a regulatory region. It helps to regulate how the DNA is used. Okay, so the quick recap of this then. So we've got surprisingly little of our DNA is a gene coding region, only about 1.5% of our DNA. And we have about 21,000 genes, leading to roughly 21,000 proteins. So about 3% might be regulatory, switches that help to switch on and off expression of genes and control development. We have quite a large non-coding region. In different organisms, it might be 5 up to 45% of the total genome, and it doesn't code for a protein. They're often made of highly repetitive sequences, 5 to 300 bases. For example, the longer ones, up hundreds, 10 up to hundreds, are VNTRs, and we talked about the STRs, the short tandem repeats, and they're also referred to as satellite DNA regions. And these are the parts that we use in DNA fingerprinting and profiling, and we'll have a look at why. So overall, what is the function of these non-coding regions? We have regions in between our genes which don't code for a gene itself, for a protein itself. And so these regions nearby, they can have a role in helping to either act as a positive thing that causes more of that gene to be used, or a negative thing. So it's a code which tells the machinery in the cell either make more of this. So for this gene here, the first one, we've got something which is acting to increase. And this could be termed as an enhancer. It's causing more of this gene nearby to be used and transcribed into protein. Whereas over here, we've got a negative. This could be called a silencer. And what it's going to do is work to maybe decrease the transcription of this gene. The other thing is that also we have bits, and we could consider them to be part of the gene, but they are non-coding in that they don't create a protein. There's a bit next to every gene, so let's just do that as a little green box. Next to every gene is a sequence which is called the promoter. And what this does is it causes transcription. We're going to see some of the enzymes involved, but there's an enzyme which needs to come along and it needs to bind. So I've just drawn this circle here as the enzyme. There's an enzyme which is going to run along the DNA and use it. And so for this enzyme to bind here, it needs to recognize a promoter. It will bind to that promoter. So some of the regions are promoter, which causes transcription. It allows the enzymes involved in transcription to bind. So as a quick recap, the non-coding regions could contain promoters, which are required for gene expression, for using the DNA to make mRNA in transcription. There could be silencing and enhancing sequences which either reduce the use of the gene or increase it. And so we can say that these regions are regulatory in function. They're also structural because if we remove it, removal leads to loss of function. So they've done experiments cutting out these regions and just sticking back together the genes, and then it just doesn't work. And we said they're repetitive sequences. So how can we use these repetitive sequences in DNA fingerprinting? Well, a DNA fingerprint, it was called a fingerprint because fingerprints are unique to individuals. There's very little overlap. And so this is a genetic element that's unique. So if we look at different people, we will see that they have different numbers of lengths of repeat. So person number, sample 3 here, will have a longer section. And if we were to kind of draw these out in terms of the lengths of these sections, we've got different length sections depending on the person, 1, 2 or 3. And so what they do with DNA fingerprinting is they use enzymes to cut. It cuts at a particular point. It will recognize a particular sequence, and it's like a pair of scissors. So by cutting, the cutting leads to different lengths of DNA in different people because they have different numbers of repeats. And then what they can do is they can separate the strands using gel electrophoresis. They put these samples from each person, and a bit like chromatography, all the samples start at one point and they run them out. And we can think of this as being kind of like the spots which are on chromatography. So we can see down here that when we run them out, they will have banding in different places because these different sections will have different lengths. And so a different banding pattern due to your different number of repeats in satellite DNA will lead to a very, very low chance that you will get exactly the same bands. So we're showing multiple bands here, so it's actually different enzymes that they use. They're not just cutting this one, this particular sequence. So different enzymes will lead to different banding pattern. And so if your banding pattern matches, then it means that it's the same, you've got exactly the same number of repeats. Okay, so our recap of that was that VNTRs were short nucleotide sequences 10 to 300 bases, whereas short tandem repeats, STRs, were 1 to 6 bases long. And they are varied between individuals because the number of times that those sequences will be repeated is individual. And the different number of repeats, they are alleles, they're different forms of DNA. So different people will have different numbers of repeats at different locations, different loci. And they can be cut using what are called restriction enzymes, giving a banding pattern that we get in gel electrophoresis. Now we've gone through that pretty quick. This comes back in 3.5 to do with genetic techniques. And so this is what's called your DNA profile. Now, someone that you would match, you would have matching bands for family members. So family members would have some matching banding patterns because they have some matching alleles. But different individuals would never have all the same banding patterns because we inherit different mixes of alleles from our parents. And so this can be used in paternity cases or to identify criminals. It's about 1 in 7 million. The chances of having exactly the same banding pattern because you've got exactly the same alleles in your STRs and in your VNTRs. So the final part that we would find these repetitive sequences, which is pretty cool, is at the end, over here, at the end of the chromosome. At the end of chromosomes, there are structures called telomeres. And they've been investigated a lot because they are associated with aging. As you age, these telomeres reduce in length, and they are repetitive sequences at the end of telomeres. And they seem to have a role. They're a structural role. Because if we remove them, the chromosome doesn't work. So what their role in aging isn't quite clear, but certainly as you age, they seem to reduce in length. And that's the end of the clip.